All right, so this is talking about last week's sentence activity. So this has to deal with transcription and translation. All right, so let's check it out. So transcription is the first process. So this actually happens inside of the nucleus. And so transcription is we're turning DNA strand into an mRNA strand. So let's take a look. What does that mean? Okay, so here's our DNA strand, right? We know that DNA is composed of adenine, thymine, guanine, and cytosine. Those are 4 nucleotides, right, that we should have learned about by now. So when you made those DNA strands last week, those DNA strands were made of 4 different nucleotides: adenine, thymine, guanine, cytosine, right? And here they're always abbreviated. T for thymine, A for adenine, C for cytosine, G for guanine. Okay? So then we see that one side of so we know DNA is double-stranded, right? Here's one strand over here, and then here's another strand down here. And so it's showing us right here. Here's the DNA strand that we are going to transcribe. Okay? And so I did not do this, but I didn't tell you which ones to do. So you need to do 3 of these. Let me see here. So here's the sheet that you're actually turning in here. So you're going to do sentence 1, sentence 2, and sentence 3. Okay? And so I'm going to go ahead and do a little bit of one of them for you. So the first part we're doing is this guy right here. So this is the transcription part. Now, let me just write transcription here. So here's our transcription. Our transcription has to do with making mRNA code. And if you notice back here on these guys are in groups of 3, groups of 3, groups of 3, groups of 3, okay? So I'll go ahead and do the first couple for number 1 for you. How about that? So we start out with TAC, okay? And so when you made your DNA strand last week, there were these guys paired up, right? Adenine always pairs with thymine, and guanine always pairs with cytosine, right? So we know this. So when we go to make our mRNA here, we need to know that there's two different things. So our DNA here is double-stranded, and then we also have this nucleotide called thymine in here. But when we actually go to make mRNA, there's 2 different things. So first of all, mRNA is single-stranded, so we don't have to worry about a second strand. We're only doing 1. And then the next one is that the nucleotide, we have a different nucleotide. We actually replace thymine, and we replace that guy with uracil. Okay, so only DNA has thymine. But when we make mRNA, okay, so when you go to, oh my gosh, I got so many things to pull up on this board. So when you go to fill this guy out, there's only going to be uracil here. There's no thymine, okay? So we know that if you see adenine on your DNA strand, it will pair up with uracil, not thymine, right? And then cytosine always pairs with guanine. You know, so if this is our DNA strand and we're turning this DNA strand into the mRNA, which is what you would write down here, adenine would pair with uracil, cytosine with guanine, and then obviously if guanine was over here on our DNA strand, then guanine would then pair with cytosine here. Okay, so let's take a look at this. See if I can do this. Okay, and so my first one here. Okay, so my first one, TAC. So what pairs with TAC? What pairs with T? So we know that adenine and thymine always go together. So if this is a T, then the T must pair with an A. I don't know if you're thinking this right now, but you're like, well, hey, I thought that there's no thymine in mRNA. And you're right. This is DNA. And that's what we're doing. Again, what we're doing in transcription, we're turning this DNA into mRNA. And mRNA is the one that does not have thymine and only has uracil. Okay, so that's where that's coming from. Okay, so thymine transcribes into adenine. Adenine then transcribes into You guessed it. Uracil. And then and then cytosine, which is the next one. Cytosine pairs with guanine. All right, so there's AUG right there. So there's our first part of our code. This is our first, our 3 nucleotide base pair, okay? So then what we're going to do here, and you're going to do this all the way across. So then my next one's GGA. So what pairs with GC? All right, so I know it'd be CC. What pairs with A? Thymine. Oh, wait, we're doing mRNA, so it's actually uracil. So then it'd be CCU. And I'd keep going until I ran out of these guys, right? I'd do all of these, okay? But let's go ahead and start making this English sentence. If I'm you, when I do this, I would do all of these guys, and then I would make your sentence, okay? So then let's go over, I'm going to give you a cheat sheet. So I'm going to change this up a little bit for you. This is going to help out. So instead of sifting through this guy, so I did send this out. So here's all of our codons. So we call these 3 nucleotide base pairs codons. So we made these guys in 3 nucleotides at a time. And then what I would do is if this codon GAU showed up, so if this was a double-sided piece of paper, I'd just flip it over, but it's on my computer. So GAU is actually go, right? Well, that's a lot to flip through. It gets really confusing. I understand that. So let's get rid of that. And I'm going to give you this instead. Okay. And so how do we use this? Well, we use it like this. If my first letter in my codon sequence is A, which it is, A is the first one. I'm right here, A. So then it's going to be in this row, some here that has A, right? Because it says first base. The next one is U. So that's my second one. So I go here to U. So now I'm looking in this column. And well, it's just like a multiplication table, right? I come up from U, I come over from A, so now I know it's got to be within this box. And then how can I find it? I come over here to my third base. My third base is a G. I see G right here, and I follow this over. AUG equals start. AUG equals start. So then I'm going to go ahead and write start. Okay. Then my next one is CCU. All right. So my first base is C. So I come over here, I find C. So it's got to be in this row. My second one is also C. So I come down here in this row. So again, a multiplication table. This C comes over, this C comes up. Okay. It must be in here. And then my third one is a U. So I come over here and this is my U. CCU is I. So then I write down I. Okay, and then I'm going to slowly write a sentence. So every single sentence you're going to have, though, is going to start with start, and then every single sentence you have is going to end with stop. Okay, and that's how proteins are made. So when you're making this English sentence here, we're making a protein in your cell, right? Because these proteins go out and they do work in your body. They create your blue eyes that you have, right? They're reading these genes on the DNA. They're turning those genes by transcribing them into mRNA. And so this is your transcription. You're taking those genes and you're turning them into mRNA. That mRNA then leaves the nucleus and then goes outside of the nucleus, but it's still inside of the cell, goes to the ribosome, where the ribosome makes these proteins. And that's where translation occurs. And translation is we're translating our mRNA into an actual sentence. Or in real life, it would be actually a protein. Okay, and I think I wrote. So then next is that translation. So this is where we actually are doing the sentence making. You will translate the mRNA into a sentence. This would be a protein in real life. Okay, so you're just going to have to do 3 of those, and you're doing 1, 2, and 3. Let me know if this helps out. If it doesn't help out, let me know. And I think you're at school tomorrow anyway, so we'll get it figured out. Okay.